Welcome to the 2020 edition of the Cadiz Lecture Series and our first lecture for the new year. I'm Heather Logan. I'm the Senior Advisor in the Pharmaceutical Review Department here at Cadiz, and I have the absolute pleasure of moderating today's session and introducing our guest speaker. This is a day you probably won't forget anytime soon. But before we start that, I'd like to start by acknowledging that today's talk is being presented on the traditional and unceded territory of the Algonquin Nation. We have over 500 registrants, and I think at last count it was 513 from as far away as Brazil, but we also have people from across Canada, the United States, and Europe, so quite a large group of people online. We'll spend about 45 minutes, well, we won't, our speaker will spend about 45 minutes going through a lecture about the topic today, and then we'll open it up for questions from the audience, from in person and from those who are online. Cadiz lectures are social media friendly events, so we are encouraged to tweet them using the hashtag CadizTalks. Please do that if you're in the room, feel free. And now down to business. So today we're going to hear about an amazing thing that's happening in Canada called the Canadian Partnership for Tomorrow Project. It's a population cohort. So I'll give you a little insight, but you'll hear all the detail from our speaker. This is the largest group of volunteer research participants and one of the largest population cohorts in the world. More than 330,000 participants since June 2008, when this was established, have joined across 6 regional cohorts and 9 participating provinces. This will really, the data generated from this study will allow research to better understand and ask really pivotal questions about what causes cancer and other chronic diseases. It will help them to determine the link between the onset of illness and 2 types of risk factors, modifiable risk factors, so they're the kinds of things that we can change. How much exercise we get, what we eat every day, and non-modifiable factors such as environmental exposure. The project actually collects a number of things, including blood, urine, saliva, and biosamples. And here's a fact you don't hear every day. They actually have a Guinness World Record in the Atlantic component of this for the biggest number of toenail clippings in the world. They have 53 publications, one recently from the BC Psychiatry from the Ontario Health Study, and a range of topics based on what they've seen and analyzed so far. So lots of exciting things happening here in Canada. One of the most important men behind this work and a remarkable pan-Canadian speaker is Dr. Philip Awadallah. I'm going to have to take a pause, and I am going to have to read this off the sheet because there are so many roles and so many accomplishments of our speaker today. He's the National Scientific Director for the Canadian Partnership for Tomorrow Project. He's the Director of Computational Biology and the Executive Scientific Director of the Ontario Health Study at the Ontario Institute for Cancer Research. He's also a professor of Population and Medical Genomics at the University of Toronto, and he also was the director of the CARTaGENE Cohort project, which is part of CPTP. He obtained his doctorate in population and statistical genetics from the University of Edinburgh and was awarded an NSERC, Killam, and Wellcome Trust Fellowship to pursue his postdoctoral work. He's had faculty positions in North Carolina and the University of Montreal and at the University of Toronto. And believe it or not, in spite of all of that, he also volunteers. And I could go on, and I'd encourage you to look at it, his profile on LinkedIn and other places. It's quite remarkable. But I'd like you to hear, this isn't a talk about me, this is a talk from him, so I'd like to introduce you to Dr. Philip Awadallah, and please join me in welcoming him.
Great, thank you very much for that fabulous introduction, and it's a pleasure to be here at Cadiz. What I'm hoping to do for you today is to give you an overview of the program that was really well described, and perhaps you should be giving this presentation. That was really a great introduction to the cohort. And so what I'm hoping for you to give you today is an overview of the population cohort that we've been building here in Canada for the past over maybe even longer than 10 years now, because the thought process behind developing this cohort started in the late 90s and early 2000s. So what I'm hoping for you to get, what you're going to see today is you're going to see an overview of the population cohort, and I'm going to give you some case studies or examples of how you can use this for your science. And one of the things that I'm going to be ending this presentation with today is how you as the research community can get access to this activity as well. So why do we build these population cohorts? Let's just get started. So we like to think of these population cohorts as population laboratories. We're using the population as a way of studying who develops diseases, what factors contribute to disease, and it's very different than, if you like, a clinical cohort. Clinical cohort usually consists of participants who are are coming to a hospital, maybe it's a cardiovascular hospital, maybe it's a cancer research center or something like that, because they have a specific condition. What we're trying to do with these population cohorts is capture lots and lots and lots of information before you develop a disease. Now, that does not mean that with these population cohorts or these population laboratories that we don't bring in participants who have been diagnosed with the disease. We do. But what I'm trying to emphasize is we are trying to get a snapshot off the population when we build these population cohorts. And I'll give you some indications of why that's important for research. Now, this next slide I usually use is a bit of a straw man. It's a bit perhaps unfair to Andre Picard because this article came out in the Globe in 2016. And he was making a case, and I think a very good case, for why we need to have population cohorts in Canada, right? And in major activities across the planet have put substantial investment into building the types of population cohorts that I'll be describing today. The UK Biobank is perhaps one of the most important health studies on the planet because they've made significant investments into building population cohorts to study the development as well as the underpinnings of disease. They've recruited 500,000 people. Australia's followed suit. The All of Us program in the United States is following suit. They're a little bit behind us here in Canada, so we're actually part of this community now. The US program will be recruiting 1 million people over the next number of years, and they've already started doing genome sequencing on those participants. So this type of activity is not necessarily unique to Canada, but we have some aspects because Canada is a unique population with a unique environment and a unique ethnographic distribution as well. So we need to have our own population cohort. And this is the name of the population cohort. It's called the Canadian Partnership for Tomorrow Project. It is a population health research platform. We do not just support population health research, we support broad areas of research, but because it is population-based, we call it a population health research platform. And it has already recruited and consented over 320,000 participants from 8 provinces, in fact. The 9th province, which was Manitoba, just came online, and they are broken up into 6 cohorts. And that's because what we've done is we've tried to manage this activity, because our healthcare systems are largely led provincially, through the different provinces. So while I am the National Scientific Director and I lead this activity out of the University of Toronto and the Ontario Institute for Cancer Research, we have important regional scientific directors in many of these provinces and populations and provincial cohorts. 6 cohorts, the Atlantic provinces were all put into one activity, so all 4 provinces there are part of the activity. Manitoba, as I may have mentioned, just came online, so we've just begun our recruitment activities there. And you're seeing the names here of these population cohorts here. So Manitoba, I'm highlighting them because we're helping to get them out of the door in terms of the recruitment, has just come online and is being driven out of activities out of Cancer Care Manitoba. And these are, as I mentioned before, the regional activities that we have supporting the CPTP or the Canadian Partnership. As was mentioned during the introduction, I was the leader of the CARTaGENE activity for many years, so I was involved in the recruitment of the 40,000 participants that were consented and recruited from 2009 to 2015. I'm now leading the Ontario Health Study as well as leading the National Scientific activity as well. So those are the numbers, and those are how they're broken down. There is now a national office. It's now at the University of Toronto in the School of Public Health. A very quick overview of what we managed to pull in terms of the population cohort. As I mentioned before, over 320,000 participants. In fact, our numbers are actually right at 333,000. That's a very easy number for me to remember. That includes 1 in 60 Canadians. Actually a really interesting number, right? That number of individuals in Canada have believe that this type of activity is important. And when I say 1 in 60 Canadians, it's 1 in 6, because the population is over 33 million, that's 1 in 60 Canadians within the age demographic, right? So within the age ranges of 30 to 69, 30 to 79, were recruited. So again, that's quite a substantial number. It's a very interesting number, and I am always kind of impressed by what is it that captures the imagination when we're trying to do these types of activities. It is a longitudinal population cohort, meaning that we have consent to follow our participants over the next 30 to 50 years. right? And longitudinal activities include a number of things. They could mean us reaching out back to the participants and asking them to fill out questionnaires, whether it's around physical activity, diet, residential occupational histories come into play. But also we have consent from each one of our participants to link to administrative health records. And that's really important in terms of longitudinal follow-up. It's one that also allows us to focus in on those types of questionnaires in terms of to ask kinds of questions that you wouldn't get from an administrative health record, right? We wouldn't necessarily capture information about your diet, right? We can capture information about blood sugar from those types of information, but not those types of other behavioral patterns. So we can consent and link, we have consented and linked to administrative health records, and you're going to hear about how we can use that for some of our science later on in this presentation. Physical measures participants were invited to the clinical sites across Canada, so we have physical measures on a number of our participants. That includes things like blood pressure. People were invited to complete other types of activities like grip strength, heel ultrasounds. From a number of participants, we've captured MRIs. From about 10,000 participants across Canada, they've actually completed an MRI evaluation or scan. And we've got biologics in a number of these participants as well. And that supports a lot of the activities that you're going to be seeing as well, particularly genetics. And so there's some highlights here as well of publications which have appeared that have used some of this information. At present, we've supported at least about 80 projects thus far. That's either using the national data or data coming from each of the regional activities. And so you're going to hear about some of that science in a moment. So these are the kinds of information we're capturing on some of these participants. So as we said, we get biologics from each of our participants. Of course, that supports genetics, but we capture other information from those biologics. Blood sugar, for example, is one thing I mentioned. Lipid levels, cell counts you're going to hear about as well. Serum and plasma. All of this material is stored in a biobank and fractioned in a way because one of the things that we support is releasing biological samples back to the research community so they can use it for their science. That differs from a number of other cohorts across the planet. We wanted to make sure that we were able to support and be forward thinking because technologies change, and we wanted to make sure we have the samples in the biobank so that if somebody has a new technology they want to test, that sample is there. These are some of the physical measures. We're not going to get into, I'm not going to read off all of the physical measures. And we also capture environmental measures, and we're going to hear a lot about environment in this presentation as well. So, one of the things that I spend a lot of my time doing is working in that partnership space. So CPTP has partnership as part of its name, and that's important because we're not just partnerships internally, partnerships across the provinces, but we're also partnering with activities such that we can enrich our cohort through building into our cohort information coming from other activities. So we, as I mentioned before, we're trying to build the cohort so that we get information about administrative health questionnaire information, ICD codes, that kind of information. But we're also trying to enrich the data so that we get environmental information. From each one of our participants, they've given us a 6-digit postal code. And that helps us actually find information about that participant in terms of the kinds of things that they are exposed to. They also tell us about the drugs they take, which is of course interesting from organizations like this' perspective, so that we get in a sense to some extent about how effective those drugs are. We would like to know how many drugs are you taking for glycemia, for example. How effective are they? How does that impact, say, genetics, gene expression? Does genetics play a role in how the efficacy of those drugs? So as I mentioned before, we spend a lot of our time building those partnerships, and that helps us enrich the cohort so that we learn as much about our participants as they age, so that we can understand what kind of diseases they develop and why they develop those diseases. And that allows us to support studies in both a retrospective manner as well as in a prospective manner as well. One of the things I also want to emphasize is, and I think I did this at the beginning of our presentation today, was that we are not alone in this activity internationally. But now that we do have a population cohort of 300,000 participants, we now belong to a fairly exclusive international club, if you like. And these are some of the activities that are already out there. There's a new consortium that the CPTP leadership or myself are involved in. I'm on the steering committee of something called the International 100,000 Cohort Consortium. It was quite interesting actually building this consortium to actually identify and find how many of these cohorts actually exist that have over 100,000 participants. Some of them are not your traditional type of cohort. The first one in this list is 23andMe, which is actually a commercial activity. You could also throw up Ancestry.com if you like. I was on the advisory board with them for many years. They have recruited lots of information about participants, not just about their genetics, but also about health. But some of these other cohorts have been out of the gate for some years now. The EPIC cohort, for example, has been around since 1990. That is actually a pan-European activity. The UK Biobank I already mentioned, which is a UK activity, but right there in the middle is the Canadian Partnership for Tomorrow project. All right, so what is it that we've captured? So there is a marker paper now. It appeared in CMAJ that describes the Canadian Partnership for Tomorrow project cohort, and we captured information about things like overall perception of health in the cohort. So this is the kind of thing that we've captured and described in the paper, and this is the kind of information we can give back to the research community as well. I'll show you how you get access to that kind of information. We've got information about self-declared diagnoses. Cancers there at the bottom. Around 30,000 participants in the CPTP have been diagnosed with a cancer. About 70,000 participants have been told that they are hypertensive. We spend an awful lot of time, if you like, checking whether those self-reported conditions are accurate, whether they match up with regard to a actual clinical diagnosis by comparing it to the administrative health data. So for example, the cancer data in particular is actually really good, in fact, with regard to the match between self-reported and finding something in, say, the Cancer Care Ontario registries is the kind of, give you an example of the kind of thing that we can compare to. The same thing with BC Cancer Agency. So we have to do that provincially. In some cases, we can do that nationally. So as I mentioned before, we've been spending an awful time in the space where we're trying to do linkages, not just with regard to administrative healthcare data, but also trying to link to environmental records and so on. So I'm going to use that to set up a case study for how you can use this information for some science. And now, oops, did I go forward or went backwards? All right. One activity that we've been spending an awful lot of time working with is the Canoe project. So Canoe stands for Canadian Urban Environmental Health Research Consortium. Canoe is a great name for an activity like that. And in this activity, it's a CIHR-funded activity, initially started by Jeff Brook with a number of, it's still led by Jeff Brook. He's the PI on this activity with a number of us as co-PIs on this activity. It's a substantial activity to capture as much environmental exposure information from various records across Canada. And we've been spending time linking that information to CPTP participants. And so this is where that 6-digit postal code that I mentioned before becomes in really handy, because we can use that 6-digit postal code to say something about what PM 2.5's particulate matter 2.5 looks like where you live. Some things with sulfur dioxide, green space, walkability indices, your accessibility to food, and see how that impacts things like health and how that potentially might impact with your genetics. So that's what we're going to hear about in the next 15 to 20 minutes. So hopefully you might find that interesting. So in every location in Canada, it can be described by a complex set of environmental factors. There is a Canoe website. So if you do a search for Canoe, it's not that hard to find, in fact. You can actually capture this kind of information. You don't have to go through the CPTP. It has a standalone website, so it's great. And we've been using that information to look at things like how what you're exposed to interacting with your genetics might impact all of these things in between and have an impact on a phenotype. Okay? And what I mean by phenotype, I don't just mean disease outcome. Right? That's obviously an important phenotypic trait, right? But there are a number of things that happen to you before something develops in terms of a disease as well. So genetic variation might be impacted by or interacts with your genetics in terms of impacting how genes are turned on, how they impact the proteome, the metabolome, and how they interact with, say, things like blood sugars, which might then impact, that's just an example, but how that might impact, say, a disease outcome. Okay? So something that's out there now is a term called the exposome. So you've heard about genomes and you've heard about metabolomes and proteomes. There's something called an exposome. Exposomes are pretty much anything that you're exposed to. It could be air pollution, they could be how much stress you're subjected to, the drugs you take. All of those things will have an impact on your overall health and how your body responds to it, and even how your genetics may respond to it. So we've published a number of things in this space, and I'm going to describe some of these things. I'm not going to spend too much time on those articles. I'd rather get right into the science of this. And everything you're going to see for the next number of slides now was done in Quebec, but is now being funded to be explored across Canada. So we started in Quebec because Quebec was actually one of the first cohorts out of the gate, and it's actually a really interesting segment of the population from a genetics perspective. As most of us know, Quebec was founded by the French about 400 or 500 years ago, and so there's a French-Canadian, if you like, genetic repertoire of mutational variability in that population that is unique on the planet, and speaks again to why we need to have a Canadian cohort, because there are a number of unique Canadian populations. This founding population has had an impact in terms of how that founding event has had an impact in terms of how genetic diversity is distributed. And so one of the things that we first started doing in Quebec is started genotyping individuals in this population. And so that's what you're seeing here in this slide. So on the left, you've got a very simple principal component analysis of genotypes just from individuals from, on the right, Montreal, Quebec, and a place called Lac-Saint-Jean-Saguenay. Now, we have them colored there on the left, and what you can see, and I was really surprised when we first did this analysis. This data is already a little dated, comes from around 2012, 2013, but I was really impressed at how well I could separate individuals from these three cities. When I came to Quebec, I thought French-Canadian genetics was largely just French-Canadian genetics, but this genotyping at this resolution actually was able to discriminate peoples from these three different cities, which probably, and then I'm doing a bit more research and probably shouldn't have been so surprised, is this actually reflects some history, of course, or anthropology, for lack of a better term, right? That there are sub-founding populations in Quebec, and this is actually, your genetics is actually capturing that information. So your genotyping actually allows me, or the genotyping I've done, allows me to identify actually where historically or ancestrally you came from within Quebec as well, not that you're just French-Canadian. And you can see that I've actually been able to map that at a 3-digit postal code level here on the map on the right. Now, I say that was surprising to me. Since then, when you go to, say, places like 23andMe and Ancestry.com, and they present at the American Society of Human Genetics meetings or so on, you'll see actually these beautiful maps, even of just, say, the United States, where you can see really interesting pockets of genetic diversity as is associated with colonization of the Americas. So that's information that's now coming to the fore. I'm not going to spend too much time on that, but on identity by descent, that's a population genetic term. But what I want to say next is that we can see this in terms of genotypes, but what we can also see is a similar geographic cline or variation associated with how genes are turned on or off. So we call that a transcriptomic line, transcripts, gene transcripts, and that genetic variation that we saw by region, we can actually see similar patterning when we look at how genes are turned on or off in terms of gene expression, and that's that transcriptomic line. So it's really quite fascinating that geography is recapitulating not just genetics, but how genes are turned on or off, which then led us to a very important question that's at the foundation of almost anything we're doing as geneticists on the planet. Is that kind of variation in terms of how genes are turned on or off, is that driven by your underlying genetics, the genetic variation, or by environment, right? It's the classic question in that space. So G by E, that's the interaction, which is why I'm here today, I think, in giving this presentation. So in general, we ask questions like, how much of the variability did we see in gene expression? So this is just numbers, of course, but this is an analysis of variance, and so we ask things in terms of how much of the gene expression variation can be explained by things like gender, cell counts, the region from which I sampled the blood, and so on. And one of the things that we saw was that region was largely defining that gene expression variation. Now, I'm not going to spend too much time on this plot. I'm going to spend a little bit of time because it's really important. Because what I think we're able to see, and this is what these plots tell us, is that where you live drives gene expression variation more than more than your genetics. Okay, so these plots are called volcano plots because they look like volcano explosions. And so what we did was, because I was able to say from your genotype where you're from, right? So I can say from your genotype where you're from that you were ancestrally either from Montreal or ancestrally from Quebec. I can then ask, if you're now living in Montreal, whether your gene expression profile matches that of somebody who's ancestrally from Montreal versus living in Montreal. Okay, so that's an important distinction to make, because I can tell based on your genotype where your ancestry is from and compare that to where you're actually living today. So what you see there on the top figure A is that you get these big explosions. There's lots of variability or lots of variance when you're comparing somebody from Montreal versus somebody from Quebec. But if you're looking at, say, at the bottom in figure, say, C here, Montreal locals versus, say, Quebec migrants, so historically or ancestrally from Quebec, but now living in Montreal, you don't see that major change or difference in gene expression. So what that says is that genetically you may have been from Quebec, but now that you're living in Montreal, your gene expression is actually matching where you live. So it says something about where you're living is impacting how your genes are being turned on or off in terms of exposures. Right? There's something in Montreal or something in Quebec or something in Lac-Saint-Jean that is driving your phenotype. If gene expression is a phenotype, that's what that is telling us. So what is it about these regions, right? So that's the next question. So that comes back to linking back to Canoe, right? And looking at those 3-digit postal codes and asking questions about what is different between people in terms of where you're living, in terms of Montreal, Quebec, Lac-Saint-Jean, Saguenay. And so these are the kinds of things that we're able to capture, and as I said, is in the Canoe site. And so things like ozone, nitrogen dioxide, that's the French writing there, sulfur dioxide, those are the things that seem to be the most different, and in fact at higher prevalence in places like Lac-Saint-Jean, Saguenay compared to Montreal. And we can do this at even finer resolution. It's not that Lac-Saint-Jean, Saguenay itself was sticking out. Because we have 3-digit postal codes, I can look at, say, North Shore, South Shore, Montreal, and ask if the patterns of pollution, for example, or exposures are similar there at that level compared to, say, Lac-Saint-Jean, Saguenay. And in fact, we can actually say, I'm picking at high resolution geography here, but we can actually make those types of claims and statements as well. So that was published in Nature Communications not that long ago, in fact, about a year ago, and that got us some activity. So we actually did appear in the Globe and Mail finally in a good kind of way, not just that we haven't been doing work. So they're now aware of us, or somebody at least in the Globe and Mail is. And now, so any apologies to anyone, of course, at the Globe and Mail. You're all very lovely people. So. One of the things that I'm not going to spend too much time on, this is called an EQTL plot. This is the kind of thing that geneticists now do, and it's kind of de rigueur in the community. But what we try to do now is we can actually map a genetic variant that we actually think interacts specifically with an exposure. And that's what this map is. So you see you have this big circle of 1 to 22. That's 1 to 22 chromosomes. The sex chromosomes are not included here. And each one of these dots in the middle are positions that we think are linked statistically to how genes are turned on or off and interacting with an exposure. That's what that is. So that's really kind of how some of this field is now moving in terms of trying to understand how exposures interact with your genome, particularly those parts of your genome which are sort of the dark matter, if you like, the non-coding regions of the genome, which aren't specifically genes but might be regulating how genes are turned off. Lastly, in this kind of space, I wanted to talk very quickly about some of the genes that we're capturing in these types of studies. So, of course, when you're looking at, air pollution, it's not surprising that the genes that we're capturing are seeing an association in terms of variation with regard to differential gene expression associated with things like oxygen transport, inflammation processes, and so on. So it tells us something about we're probably working in the right direction here. So as I mentioned before, this is just a quick summary slide. We are doing this kind of type of work and now expanding it across not just Quebec, but Canada as well. So we have some CIHR funding for that type of activity. Okay, so that's one type of case study for the kinds of things that we can now do with a population cohort. So everything I've just done is being done in the population sort of context, right? It's not a clinical context. Some of these people have been since diagnosed with the disease, but everything I was able to do because I've been able to manage and recruit individuals from the population. Another case study I want to support or to emphasize are the kinds of things that we're now doing, thinking about an aging cohort, because this is an aging cohort. These participants came into this population cohort about 2010. I'm getting old with the population cohort. That's when I first started coming into the cohort. And so what we have is a number of participants who've come in at varying age ranges, 30 to 79. And we're evaluating what are the major factors that are associated, both genetics as well as environment, that are associated with developing diseases associated with aging. So I've just got here a plot. This is not a plot from us. This is just something published in terms of the frequencies of things like hypertension, diabetes, cancers. Our numbers, in fact, in the CPTP reflect what we capture in StatCan for those numbers. So even though I never want to say that we're terribly representative of the population cohort from a statistical perspective, we actually do pretty well. We're in terms of capturing the right numbers of frequencies of diseases. And so, one of the things that we know about in terms of the kinds of things that are associated with aging are lots of genomic factors that might be associated with change over time. And so everything I was just talking about in the previous set of slides was looking at germline or heritable factors. But one of the things that we can do with a population cohort is also not just look at the heritable factors, but how your genomes change with time. Right? It is not a static, singular activity. One of the things what we've tried to, and when I started this activity, I didn't think the technology would even be here by this point, that we could look at how your genomes might change, how you might start accruing mutations as you age, as you're exposed to things in terms of lifestyles, the things that you do normally in your normal regular day life. But these are the types of things that contribute to your age. And this actually comes back from a Cell paper called The Hallmarks of Aging. And one of the things that we can do, because we have a lot of it, is look at how your blood ages with time. Blood is a dynamic tissue because we have a lot of it. We have a lot of it from all of our participants. So we can actually do some really interesting science here. We're doing follow-up studies of blood samples. And we can look at how your blood changes. Now, blood is a dynamic tissue. It starts off with hematopoietic stem cells. It leads to these progenitors that are myeloid or lymphoid, and that lead to other important blood types associated with immunity and just everything that's necessary for life. And so as you age, your blood constitution changes. It actually becomes more myeloid biased. And we think we now know why why that happens. That's some great research actually being driven out of by the folks out of Princess Margaret, for example. John Dick is leading the charge here, studying things like age-related clonal hematopoiesis. There are genetic factors that might be associated with those changes as well. And we actually know that there are thousands of heritable variants associated with why people have varying counts of blood. And this is one of the best actually examples of heritable variation. And so we've been studying that in the context of what contributes to, say, healthy aging of blood. We know that there's a lot to be done in terms of the science of why people develop diseases, why do people develop abnormalities in blood, or anomalies, or malignancies. But we're interested also in who are those individuals who age really well. And in our cohort, these people are starting to become super agers, right? So those are the nonagenarians or the centenarians who have perhaps a quality of life that's, or if you like, blood phenotypes in our case, or health phenotypes similar to, say, a 50-year-old, right? So we're able, because we have 300,000 participants, we can actually stratify our population cohort and identify who those people are. So I have one student working on that, actually a number of students now, working on that particular activity, and she's been looking at the data coming from the CPTP as well as the UK Biobank. As I mentioned before, we have over 300,000 participants in the CPTP. UK Biobank has done a fantastic job of releasing data to researchers as well, and if you like, they are the tip of the spear in terms of making sure data and information is available to the research community. And we've been looking at CPTP and UK Biobank in terms of questionnaire data, physical measures, cell counts, as I mentioned before, for all of the CPTP participants. Many of them. We have information on cell counts as well, so we can look at quality of cell counts and how that impacts health. And what we've done so far is Alyssa Bader, who's one of my fourth-year PhD students, has been looking at some of the data in CPTP and has developed a risk score that seems to be very nicely predictive. And actually, we've been testing that risk score with longitudinal administrative data that we've been capturing in terms of how predictive is it of defining somebody's health. And we've actually just stratified the participants. So that's all this figure is here. So one of the things is we've taken Alyssa's risk score, the CBC risk score, based entirely on cell count information. It ranges from 0 to 10 on the x-axis. On the y-axis, just proportion of participants. In this bar plot, you've got ages of our participants. And that allows us, stratifying the cohort, to identify say OH, which are old healthy people. It's a terrible name, but I don't know if I'm in that group, but hopefully I am. The old healthy participants, and allows us to compare your blood profiles of old healthy versus old unhealthy. Allows us to compare old healthy versus young healthy. What is it about your blood profile that actually keeps you in that old healthy category? Is it similar to a young healthy category or blood profile? Or does it actually look different? Is there something different about your old healthy category that's different young healthy that keeps you up there? And then we look at old unhealthy just to kind of round this out to compare to young unhealthy as well. And it allows us to test hypotheses, like are there protective mechanisms in old healthy blood different from old unhealthy, or are there healthy aging mechanisms in old healthy blood that are similar to that in young healthy as well. And I'm not going to spend too much time here getting into the genetics, but slide's supposed to move forward. Oh, sorry. One of the things that we are capturing, for example, this is just how we've defined individuals, is that what we tend to see in those individuals that are either young unhealthy or old unhealthy is that variance in cell quality or blood CBC quality kind of starts to explode, right? And that's what you're seeing there in those figures there. So RDWCV is red blood cell coefficient of variation width. So red blood cells get the variation in cell widths get very large in both the old unhealthy and young unhealthy. White blood cell counts, variance starts to increase as well. And that might be due to a number of factors. It's not abundantly clear why that's happening. In fact, it could be that there are, say, immature or progenitor cells that are that haven't matured properly that are impacting, say, the size of these cells or their counts as well. That's the stuff we're interested in, and that's why we started pulling these samples out of the freezers and starting to explore what genes are being turned on or off, how are they mutating, why these genes are turned on or off, and what genes are being turned on or off. And so this is just something of an advertisement for the kinds of things that we are now able to do with the CPTP, and that the samples themselves have actually been stored in a way, and this kind of comes back to what I was saying earlier, have been stored in a way that we can actually do or apply some of these really novel new technologies that have come to the fore, single-cell technologies. And so what you see here is, again, something of a principal component plot. This is actually a UMAP plot, but each point on this plot is the single-cell RNA sequencing signature of a cell, right? So we are able to now capture that information here and find these clusters, and they're just here listed as clusters 1 through, I think, 20, and use the information about which genes are turned on or off in these cells to identify which cell types they are, right? So then we can classify them as B cells, CD4 type cells, T cells, dendritic cells, etc., right? And then ask those types of questions that I was asking before. Which of these cell types are different, beyond just looking at the CBCs, but actually looking at this type of information to see which of these cell types may be differing between the different 4 groups that I had identified earlier, and identify maybe also which genes might be acting in a different way in those different groups as well. So for example, B cells, here just some really crude kind of comparisons, in the young unhealthy and old unhealthy are very similar in terms of their frequencies. Proportions in old healthy are also similar to young healthy as well. And that might speak to a young healthy aging phenotype. But one thing that I thought was really interesting was that in our old healthy group, there were a class of cells that we are not yet able to identify with this technology. So I don't know really what's going on here. If this is a new cell type, this is what these new technologies are being able to identify as well, is that our canonical cell morphologies or phenotypes are actually starting to change, right? Because we're now actually capturing things at a greater resolution. But in our old healthy group, we've got this elevated proportion of what this category that we don't know what they are, of cell types, so this unknown group. So this is really quite fascinating or interesting. Not going to, again, spend too much time on this, but we're then now looking at the individual gene expression signatures, so say in CD8 T cells, trying to identify protective genes, comparing old healthy versus old unhealthy. Similarly, we're trying to find healthy aging genes as well that might be similar, say, between young healthy and old healthy, but are different between old healthy and old unhealthy individuals as well. Again, it speaks to the kinds of things that we are able to now do with the samples in the biobank, having stratified our individuals. And one thing I want to emphasize is that we're collecting all this information. This is all going back into the cohort. Anytime anybody does these type of experiments, the cohort gets further enriched with this type of information. So any genomics that's being done, we have policies that say it has to go back and enrich the cohort and be made publicly available to individuals. All right, so the last case study I'm going to be spending a bit of time just emphasizes what we can do with a population cohort where participants have consented to allowing us to let them, allowing us to link their administrative health data to our cohort. And so one of the things that we're focusing on is work with regard to cancers. The cancer registries in Canada are actually extremely very well up to date and useful, with many great people working on them, largely at the provincial level. The Cancer Care Ontario, which is now a different organization, has the Ontario Cancer Registry that they spend a lot of time working with. And so one of the things that we've been doing in Ontario is linking the Ontario Cancer Registry to the Ontario Health Study participants. Everything we're doing in Ontario is also being done in BC, in Alberta, Quebec, and the other provinces as well. In fact, because the BC cohort itself, for example, same thing with Alberta, is actually the participants were recruited as part of BC Cancer Agency or part of Alberta Health Services. Those linkages are actually quite easy to do. And it's actually, you can very quickly get information from those cohorts about, say, numbers of participants who have developed a cancer. So one of the things that we can do, so I'm going to focus here in Ontario just because I've been spending more of my time with the Ontario stuff with regard to some of my own personal research, is identify in the cohort who came into the cohort, say, healthy, and then has been since diagnosed with the cancer. And then we call those incident cases, right? So what we then might be able to then track is, say, if somebody's participant has joined at a particular time point, maybe they again gave us a blood sample, and then since then they've been diagnosed with the cancer, right? So that is really fascinating and fabulous information to have, because you're capturing information that wouldn't again be captured in a clinical cohort, because that person wouldn't have had any reason to give a blood sample or a biopsy unless, you know, they were part of a study like this. And actually, one of the things that's really been interesting is, as I give these types of talks, I was at a Terry Fox Marathon of Hope workshop the other day, and clinicians were coming in to me and say, hey, listen, we got participants coming into our cohort or into our clinical trial studies, and they've said to us, hey, we took part in CARTaGENE in Quebec. Is there a way that my CARTaGENE data could be linked to the clinical trial information? So one of the things that we need to be doing now in the future is actually breaking down some of the silos that we have in terms of research activities, so that we can better link up some of the data sets or data sets such that we can identify who's got what in various cohorts across Canada. And again, with this linkage data, we can also, as I said before, validate when somebody says they've been diagnosed with the cancer. We call those prevalent cancers. And so this is the kind of information we can capture and pull, and now have and seen in Ontario. And I think these are for 2015 data, so this data might actually be a bit dated. And so although the Ontario Cancer Registry is a great resource, anything we do is going to be 2 years. There's going to be a bit of lag of 2 years. So data from 2019 that we've had, well, it's now 2020, but 2019 was our last request, will be information with the Ontario Cancer Registry. Information will be up to 2017 with respect to diagnoses, right? So anyway, with regard to that, I think this might actually be 2015. These are some information that we have for those individuals who have come into the cohort, provided a blood sample, and been diagnosed with the cancer. And so we can map when, so that's age of the individual on the x-axis, and then the time to diagnosis on the y-axis as well. So that allows us to do some interesting science. What we see, of course, is that rates of accumulation of incidence cancers and prevalent cancers are going up, but also allows us to support some really interesting science with regard to how blood ages, right? And can we use blood going back in time to identify factors factors that might be associated with a diagnosis prior to or previous to a traditional diagnosis, right? So this is some work that we've been very happy to be part of with John Dick and his colleagues. And I mentioned, I think, this process before. This process, this figure that I'm showing up here is, you can't now go to a genetics conference or a hematology conference without hearing about age-related clonal hematopoiesis. Age-related clonal hematopoiesis is this process that I'm talking about here, where mutations accrue in hematopoietic stem cells, and they are a precondition for developing leukemias, but they also might be associated with the development of many other phenotypes, including cardiovascular diseases. And that's one of the reasons why it's such a topical area of research, is that these mutations in blood are not just associated with blood malignancies, but might also be associated with many other morbidities as well. So John was critical in identifying some of these mutations, and this is actually just a figure from colleagues and collaborators at Harvard. Ben Ebert's team have been spending a lot of time also associating these mutations with cardiovascular disease. And as I said before, the driver mutations that are accruing in hematopoietic stem cells in blood are clearly important with regard to leukemia, but they don't always result in leukemia. As I said, sometimes they have no phenotypic effect, and we're interested in why that happens, but sometimes they also have an association with another phenotype like cardiovascular disease. So John's team published this paper that the folks at OICR, my organization, were very happy to be part of, where we were able to capture events. This was a paper that was published in Nature 2 years ago now, where when we go back into our population cohort, we pull blood samples from individuals that have been known to be diagnosed with a cancer, but when they came into the cohort were healthy. We can see that there are early signatures of those mutations in those blood samples, and those early signatures are almost 10 years prior to the diagnosis of AML, or acute myeloid leukemia. So again, another really great emphasis for why these population cohorts can be valuable. And so a lot of our research at the Ontario Institute for Cancer Research is exploiting that type of utilization, where we have many blood samples from people who didn't develop a diagnosis or didn't develop a disease when they came in, and trying to identify or develop new biomarkers for early detection. And so that's pretty much the end of the science of this talk. I just want to mention some other projects that have come in and utilizing the CPTP. Sonia Anand was the person driving the MRI program from the CPTP, and she's been publishing many articles using the MRI data from the CPTP cohort. Isabelle Fortier, I want to highlight as well, has been critical in making sure everything you're seeing in the CPTP across Canada is harmonized. What you're capturing in Alberta is similar to what you're capturing in Prince Edward Island. Okay? Sonia's project is called the Canadian Alliance for Healthy Hearts and Mind project. It also included the recruitment of another 1,500 participants from Indigenous populations. One of the things that we are quite excited about is, because the CPTP is so large, it really captures the fabric of the nation with regard to its ethnography. We have 7,000 participants who have been self-declared Indigenous participants who consented to the program. And I would not have known that if we hadn't actually captured information about ethnicity. But it's really interesting to see that community is interested in these types of projects. And when we go back to Indigenous community leaders, they say, this is fantastic, because felt like we've been left out of the precision health, precision medicine discussion. There are those individuals there who have already consented to this activity as well. So that's what I wanted to highlight with respect to what the cohort has. This is how you can get into it. It has a portal. It's not the best portal. We're working on updating it. It's good enough for now. This is called the portal.partnershipfortomorrow.ca. This is how you can find information about where data is, which is all it sits at the Ontario Institute for Cancer Research, what kind of biosamples we have, plasma, serum, urine samples, or so on. We heard about toenails. I'm not sure I'm that proud of the toenail thing, but it's a thing, and it's obviously very important with regard to exposure research. We do have an access process as well. The forms are up there as well. You can make access for data. You don't have to have biosamples in your access request, but you can make access requests for biosamples as well. We do have this office. Best to have an REB before you come in. but it's not. You do definitely need to have an REB when if you're going to get data from us as well, especially if you're going to get biosamples. So, I want to thank you for listening and thank the participants in the Tomorrow Project as well. Many funders that have supported this activity as well. They are highlighted here. It is funded by the Canadian Partnership Against Cancer, but we have many regional funders as well, and and it is a partnership with all of these organizations and activities. So I'd like to end there and just say thank you very much for listening.
Thank you very much, Dr. Awadallah. So we have time now for questions. We're scheduled to end in about 25 minutes. We will take as many questions as possible from those of you in the room and those of you who are online. So if you're in the room, just the simple raise your hand. When I call on you, make sure you don't do what I did and make sure you turn your microphone on so everybody online can hear your question. Please also identify yourself, your name and your organization. If you're online, there are two ways to submit questions. You can type your question into the appropriate box in your web interface, or Peter, the email to send questions to? events@cadith.ca. events@cadith.ca. So that's how you submit your questions. And so as people are starting to think about the questions that they want to ask, and I don't know if you'd like to take questions here and I can moderate from the chair, but I do have a couple of questions, so I'm going to pause and see if right now there's any in the room, and if not, I'll go ahead. So Jocelyn.
I'm curious, Jocelyn with Cadiz, I'm curious to know if participation rates are similar in these types of initiatives in other countries. And I'm also curious to know if our publicly funded health system makes this easier to conduct in terms of access to that administrative data, and if there are any challenges posed by a system like the American system, and also where there may be private holders of that data as well.
Fantastic. Those are all great questions. So participation rates do vary from nation to nation. Not exactly sure about UK Biobank's participation rates, but for example, things like the Estonian Biobank, which is actually one of those major, strangely enough, activities that have had very successful recruitment rates, in part, I think, because it was part of the healthcare system itself. So participants were partly recruited and consented through the Estonian national health process, if you like. So with that said, in Canada, we see varying participation rates across Canada. Quebec, we saw really astounding participation rates that were very variable depending on whether you're from the north versus from the south. So I'm not going to pick on any one particular region, but West Montreal would be very different in terms of East Montreal. Super high participation rates in the north compared to the south as well. Toronto was a challenge, right? And that has a lot to do with, I think, well, accessibility and so on. But in part, I think Quebec and places like Alberta and BC did so well. I think it's also because there was a lot of support from the healthcare systems themselves. So RAMQ in Quebec in particular was an early partner in the recruitment activity. So to some extent that helps with the psychology, I think, as well in terms of participation rates. So that speaks, I think, to your second question too, is are the healthcare systems themselves supportive with regard to linkages? Early days were a challenge. People were very risk averse. Still a lot of risk aversion out there, and that's probably important to have that sense of what the risks are and to be conscious of them. At the same time, though, you know, when you see like 1 in 60 Canadians coming in and signing up for this, that sends a message, right? And when you see people signing up for 23andMe or Ancestry.com or something, like, people want their data used for research. They want solutions wrapped around the problems. They want their diseases cured, right? And the only way that gets happen when you make many people, many researchers, make data available to many researchers so that they can do the kind of science that they need to do.
Peter, is there a question online?
There is. There's a couple. So I'll start off with this one. This is from... Sorry, I forgot to turn on my microphone. There are a couple of questions. The first one is from Paulisha Degan at Prostate Cancer Canada. She writes, "It is really impressive how many people have been recruited to this study. How representative are your participants of Canada's racial and ethnic diversity? Do you have minority groups meaningfully engaged?
So another great question. So I would say we are not as representative as we should be. We kind of get away with it because we managed to recruit 333,000 participants. So we get meaningful numbers, but in terms of actual representation, in terms of frequencies, we're not as representative as we should be in terms of our actual true ethnographic representation. There's a lot of history of that and epidemiological studies, and while that's not unusual to see that, we tend to see that underrepresented groups don't tend to engage in these types of activities. Sometimes it's a question of education, sometimes it's a question of trust. And so when we talk about actually engaging meaningfully, some of the activities that we've had to do following up in terms of enrichment of the cohort have had to actually target particular populations as well. So when we went after the MRIs, for example, we spent a lot of time in North Toronto trying to get more participants coming into the activity who wouldn't normally have coming in. And there's a different ethnic makeup in North Toronto than there might be, say, in downtown or near the lake. So that type of work has been done. More of it has to be done, particularly when we start reaching out to the thing that I mentioned at the very end of my talk, which is the Indigenous community as well. So there's a lot of conversations that are beginning to happen here as well.
Question in the room? Okay, Peter, do you want to ask the second question online?
Certainly. The next question is from Natasha at Cadiz. Is the network able to let its participants know, based on their samples they have provided, if the participant is going to develop a disease in the near future, or if the participant is going to develop a disease in the near future?
Right. So that's a challenging one, right? So that speaks to the quality that is still a research in progress, I think, in terms of where we are. And actually, this is where Cadiz actually comes in potentially handy in terms of where we are with the development of some of these tools. We do have policies in place when we know something is imminent, and we know that there are, for example, if there's a genetic mutation that is part of, say, is known to be a risk factor that we in a clinical kind of context would normally be used to identify whether somebody's at high risk. We have those policies in place, and we have a mechanism for how we inform those participants. The types of things that I was talking about with regard to research are still in research space, and we are very conscious of not being of creating too much distress. And we've learned that type of thing from, say, activities like UK Biobank. One of the things you'll hear from Rory Collins when he presents on some of the mistakes that made early on is that when MRIs were captured from participants in the UK Biobank, a lot of that information went back to either the clinician or the participant. And there was a lot of excess extra, if you like, activity that was unnecessary in terms of misdiagnosis and putting a lot of undue stress on both the system and on the participant. Now that's important to keep in mind because there is a balance in terms of how much information should go back to the participant. The All of Us program in the United States has actually been taking a very open-ended kind of program where it's almost community science to some extent, where participants can get information immediately, whether it's coming off the genome sequencers, whether it's information coming from an image, from a blood test, etc.
Okay, I'm going to take the privilege of asking my own question. Publications using CPTP data have already started to address a diverse range of health issues, from mental health status among ethnocultural minorities, blood pressure and kidney cancer risk, arsenic in drinking water and arsenic levels in toenail clippings, even glaucoma and air pollution. What's the link between those? So how broad do you see the uses for the CPTP data? Do you see any limits?
In principle, no. In principle, yes. Questions as they relate to the utilization of the CPTP are we're only going to take applications if you're asking questions around health. But that means it's fairly open-ended in that case as well. So we do ask that when researchers come in to utilize the data, it helps when there's a peer review already. So if somebody already has a grant, then we know that the science has already been evaluated by an organization. But we also have an organization within us, or a series of evaluators that will evaluate the quality of that science as well. But for example, we have data that could be used for pollsters, for example. Of course, we would never let that kind of information go, but we are interested in supporting broad ranges of health. And that's actually a challenge for an organization like CPTP, in part because we are trying to be as useful to broad ranges of sciences in different health domains as well.
Thank you. Peter, over to you for a third question.
This is from Jennifer Campbell at CIHR. She asks, how would you say CPTP differs from the CLSA? What opportunities exist for linking information between the cohorts?
So scale and scope, I think, is the first and easiest one to address that. CLSA is a great population cohort. The CLSA stands for the Canadian Longitudinal Study for Aging. It's a CIHR-funded activity. They have recruited 30,000 participants. All of those participants, recruited 50,000 participants. I don't want to misspeak on behalf of CLSA. 30,000 participants have provided a blood sample, and I think 26,000 are being targeted for genotyping. Great cohort, largely questions focused on the end of the spectrum, if you like, or the aging spectrum, in terms of diseases as they develop with regard to chronic diseases as well. There are certainly overlaps, and operationally we've tried to keep in lockstep to some extent with regard to, and we've actually spent a bit of time. One of the things that I asked for funding to support was to develop, in fact we have a portal that allows you to look at what phenotypes are actually collected by both of those cohorts, as well as other Canadian cohorts. So there are some child cohorts as well, UK Biobank, and the Swedish cohorts. And that's a project that started in 2016. Isabelle Fortier, who I mentioned, was the lead on that activity. So I think that's a really interesting sort of activity to see how you could actually build a multi-cohort scientific activity, and we're trying to extend that out beyond those Canadian cohorts and the few international cohorts to things like H3 Africa. I'm on the board for that activity, which has done a fantastic job. I'll give a plug for that. I mean, we think things are hard working among different provinces. They had to build a cohort across African countries, and actually some things are actually easier there compared to what I have to do for a living to some extent in Canada. But regardless, we're trying to bring in those types of activities so that people can then say, I'm interested in looking at blood pressure variables, and maybe there are like 5 million variables participants who have provided that kind of information. So again, back to the primary question. So we have actually looked at what kinds of data have been captured by both cohorts that are similar, so that we can move those types of activities in a more pan-Canadian kind of way.
So I was struck, Dr. Awadallah, by a comment you made at the beginning that this was something that had been talked about since the early 1990s, and it was actually launched in June 2008. So number one, that points to a long commitment to a big dream. And since it's been established and with so many opportunities to grow this platform, I think it presents a real opportunity for aspiring and established researchers in Canada and to contribute to a vibrant research community. Can you tell us a little bit about what this means to the research and scientific community in Canada? How big a deal, how much will this stimulate research in Canada, and what's it going to give us that we wouldn't have had without it?
So, to some extent, that question, I think, relates to why even having a Canadian cohort in the first place. So a Canadian cohort speaks to Canadian variability that's different in Canada, or unique in Canada, compared to what you might capture from, say, something like UK Biobank or H3 Africa, for that matter. So that allows research that, for lack of a better term, can be made in Canada or develop made in Canada solutions, right? So made in Canada solutions might mean solutions for Canadian-specific problems. CPTP, because of its size, it's 330,000 participants. It's a lot easier for moving a cohort like 330,000 participants and understanding some things that you need to understand about, say, precision health, and moving that into the healthcare system, and supporting the research that's local to get that translate that into the healthcare system, versus, say, a much smaller cohort that's, say, 10,000 or 30,000 or whatever. So one of those things that, having that kind of thing here in Canada certainly will stimulate hopefully the imaginations of our own research community, both the traditional research community as well as those that are outside what we would call the traditional setting, like new startups coming in from our young Canadian scientists, or perhaps more senior Canadian scientists, who want to look for easy accessibility for things that are relevant to what they're trying to tackle.
Great. Peter? Fourth question online. Thank you.
This is from Yi Sheng at Cadiz, who writes, I study frailty and aging among the elderly. Is frailty a research area that CPTP could assist with?
So we do have some measures like impedance. We do have some things that we think can support that type of activity. In terms of how we define frailty, of course, that relates to a number of chronic conditions. So if anything, because our cohort is now, and as our cohort gets older, it can actually really speak to that. And that also speaks to the type of phenotypes that we've already captured. So yeah, I would recommend taking a look at the types of things that we've asked of our participants, as well as the types of things that we're now asking our participants going forward with new questionnaires and so on.
Okay, Peter has another one.
This is from Nicole Mittman at Cadiz. She writes, thanks for the great presentation today, Philip. I am glad to hear the Cancer Care Ontario data linkage has worked out. Can you comment on the consent process with respect to linkages and long-term follow-up of the cohort? Is this something that you could adapt for other study designs, for example, clinical trials?
All right. So a lot of that is ongoing. So the consent process, the linkage process, so if I understand the question, I think it was about, so the consent process by itself is we had our individuals all have consented to linkages to administrative healthcare. And one way to facilitate that was participants gave us information like their insurance numbers. And where we didn't have that information, or that information was incorrect, sometimes we were able to use name, date of birth, and so on to do those linkages. To be part of CPTP, participants had to consent to that activity. So when we say we have 333,000 participants, they've all consented to that activity. That certainly allows people to drop out if they don't want to do that as well. We've had a few dropouts over time, and you expect that over time. That's normal, and so we do have mechanisms that support dropout. So as I mentioned before, we do have that in our consent. The researchers who want to have access to those linkages, this is one of those things that I'll always remember having a dinner with John Bell, who was a Regis professor at Oxford, who's a Canadian, and saying, have you solved the Canadian problem? And the Canadian problem is to some extent our provincial problem, right? Which we have a provincial healthcare system, and we have data being collected by the different provinces. CAI is great because it is a national activity, but to some extent those provincial warehouses of data can be limiting, because to some extent, if you're interested, and this is where we make a plug for why CPTP being big of 300,000 participants is important, because for those rare disorders as well as those common disorders, you need to have big numbers to capture those rare disorders. You need to be over 100,000 individuals. We've done the power analyses, but to get that information still means you have to get, at least with the linkages to administrative health records, to go to the individual provinces. It's a work in progress because the Canadian government has put aside funds and resources supporting people like Michael Schul at ICES and Kim McGrail at Pop Data BC to figure out how we can start building national warehouses, so we don't have a researcher having to go to 10 different provinces to get, say, those rare numbers of AMLs, for example, as well. But what was great about Cancer Care Ontario was we really managed to break through some barriers there in terms of getting OHS data behind the Cancer Care Ontario firewall, allowing them to do the linkages and information coming back to the Ontario Health Study and to the OICR. And I think that's the kind of thing that we could potentially take to the different provinces as well. That's great for cancer. It's not necessarily great for everything else, cardiovascular diseases, type 2 diabetes, and so on, but that's perhaps a place where we could learn from.
Yes, please. Here, press the microphone and your name and organization, please.
My name is Bryn Lander. I'm at ISED. So just to follow up on that question comment, so that I understand correctly, although you currently have participants who have consented to have their administrative health data shared as part of the research process, if someone submits a research proposal asking for administrative health data from across the provinces from the cohort as part of their research proposal, they would not be able to get that data?
No, sorry, they can get the data, but we don't have the final say, if you like. So the linkages have been done, but they still would have to get approval, and we can facilitate. And because we have partnerships now with all of those data custodians that I mentioned before, we can support the researchers in getting that information as well. Sometimes it's easier for certain conditions than for other things. And in certain places, you might have to work behind a firewall. In some places, you can get the data sent to you. Right? And sometimes it will be very research specific in terms of the type of data that's being asked for. Now, I think Nicole actually was asking as well about how can we support clinical trials. And in fact, Terry Fox Marathon of Hope, one of the reasons we were asked for that to come to that discussion or those workshops is how can we support some of the Terry Fox Marathon of Hope activities, which are largely around building some new clinical trial cohorts for follow-up studies. So they're trying to build these great what they're calling gold cohorts, and enriching those studies with as much information as possible. So we're working very closely now with them, as well as, say, those data custodians that I just mentioned before, to help support those activities.
We have one final question online.
This is from someone at Kai who wrote, from your Quebec cohort and analysis, were you able to tell how long the immigrant population were in Quebec before their gene expressions became similar to ancestral Quebecers?
So I love that question. So one of the things, so I focused on the French Canadians and the figures that I was showing before with those explosive or volcano plots, but I didn't show in that figures that we were able to do the same type of thing looking at people who were coming from West African ancestries, Eastern Europe, and so on. And this is where a bunch of my research programs start to converge, because we have, for a long time, we're working on, so I get excited about this, but we were working with African cohorts for many years. But those African samples, we were seeing gene expression profiles from so African ancestry now living in Montreal would show a Montreal gene expression profile, and an African person or ancestry living in Saguenay would look like a Saguenay person as well. So that helped really reinforce that observation that we were seeing before.
Okay, we are just about out of time. I do think there's probably a group of people who are interested in getting into the super user, super ager group, figuring out how to get into that, so maybe a follow-up talk. So I just want to say on behalf of Cadiz and all of the participants from all over Canada and internationally, thank you, Dr. Awadallah, for such an interesting talk. This is clearly an investment today based on participation of volunteers across Canada that will affect generations of Canadians to come. So what a tremendous resource for us and for Canada and for researchers. Thank you to everybody online and who's in the room today who joined us for the talk. I want to announce the next Cadiz lecture will be scheduled for March the 5th with Dr. Laureen Cipriano, the 2019 recipient of the Dr. Maurice McGregor Award for Rising Stars in the Health Technology Assessment. Registration for the 2020 Cadiz Symposium, which will be held in Toronto this year, opens next week. So make sure you've got your finger on the button ready to go. And we've supersized the event, not to be mistaken with supersized French fries, but we've supersized the event and we're adding an extra day of workshops, so it'll be even better than it has been in the past. Watch for these and details of other Cadiz events on our website. And see you next time. Thank you. Thank you. That's good.
Thanks.
Thank you.
Thank you. Cheers.
Great.